and welcome to Chapter 7, Enzymes, Energy, and Metabolism. Before we get started, I would like you to think, and you can pause the video for just 30 seconds and ask yourself, what is this, what does this word energy mean? What does that word mean? Pause. Okay, did you pause it? Well, I hope you did. Anyway, I was thinking the other day, oh boy, I've got to record this lecture about energy and what's the definition of energy? How does the textbook define it? And I thought, I kind of, you know, how you're sometimes imagining yourself doing things, and I was doing that, and I thought, well, if I had to explain energy to students, I'd just say it's the ability to do work. That's how you should think about it. And then I just double checked myself and looked in the book, and I was surprised to find, oh, well, that's how the book defines it as well. Energy is the ability to do work. So our everyday sort of definitions will serve us pretty well, except that in living things, whether it is a chemical reaction, a cell doing active transport, or an organism running a marathon, energy or work is defined a little bit differently. But energy, it's a pretty simple definition, the ability to do work. And then lastly, no, not quite lastly, secondly, metabolism. That essentially refers to all of the chemical reactions that happen in the body. And then firstly, which I skipped, enzymes. Enzymes are things that catalyze reactions, and that is one of the big topics of this chapter. How do we take A and turn it into B with as little work as possible, or with as little energy as possible? So, energy, kinetic and potential energy. So let's see here. Let's take a look at this diagram showing the bicyclist going up the hill and then going down the hill. Let's start with potential energy. Most of you probably remember this from somewhere in high school. And you know that if you're on top of the hill, and you got a bicycle, then this represents a kind of potential energy. And it's a little bit difficult to really quantify this type of energy in terms of, you know, it's not electricity. It isn't. It's not a battery, which is a difference of charge across a what is it, across a restricted flow site or something. But we know that it is a kind of energy, and we also know that it could even be used to generate actual electricity. So this is one of the things that makes the study of energy really interesting. That a person on a bicycle, and because they're on top of a hill, that could be turned into electricity. How? Well, you just put a little generator on the bicycle, and then as the bicycle tire turns, it would generate electricity from the movement of the person down the hill. Pretty neat. So let's see, that would be a kind of potential energy. And what would essentially be happening here if we wanted to go from potential energy to, let's see, to kinetic energy. So let's see, 1, and then kinetic energy is 2, and then my example, electrical energy 3. Well, essentially what would be happening is you have this potential energy on top, number 1, and then it's converted to kinetic energy in number 2. So the potential energy is being converted to kinetic energy. Kinetic energy is the energy in movement. And then 3, we could essentially like apply the brakes if we had an electrical generator set up on one of these tires, and that kinetic energy would be converted to electrical energy. That's what happens if you in hybrid electric cars. The brakes convert the kinetic energy of the car into electrical and then into chemical energy in the battery. Pretty amazing. And then starting back at the beginning here, where did this potential energy come from? As you guys probably know, energy can neither be created nor destroyed. It actually comes from somewhere. So where did it come from here? It came from energy, and that would be chemical energy. We haven't gotten to that one. I'll get to it. Chemical energy, stored in the person's muscle fibers as glycogen, glucose, and other molecules. Okay, let's see. Just another example here over near the fellow with the bow and arrow. This is also potential energy. Potential energy is a result of the position of some kind of structure where it can do work. So again, and there's that definition of work. Energy is the capacity to do work. So this setup here with the bow and arrow has the potential to do work. So that means it's energy. It's potential energy. When it is released, this turns into kinetic energy, the movement, energy of movement of the arrow. And again, that is also energy. And that would have the potential to do some other kind of work, such as maybe splitting the target into two pieces, or splitting it so the arrow can go through it. Mechanical energy. So mechanical energy is the movement of something to move another thing. That is basically what mechanical energy is. The heart is an example of mechanical energy being used to move the blood around the body. And this person's muscles, that is mechanical energy, and it is being used to lift this weight up. Chemical energy is essentially the energy between molecules, the energy between bonds. For this chapter and more the next chapter, we're going to be focusing mostly on carbon-hydrogen bonds because they have the most energy. And these bonds, carbon and hydrogen, so covalent bond here, this is chemical energy, and it is the energy that is most often used by living things to produce ATP. For some reason, this bond, carbon to hydrogen, often used to make ATP. That's where a lot of the energy comes from. As far as I know, boy, could I say all of it? I hate to say all of it. Most of it, anyway. Looking back over here, chemical energy. Here we have carbon dioxide and water. Now, the bonds in carbon dioxide and water, they don't have all that much energy, so they're not really useful to living things. Specifically, as I said, they don't have any carbon-hydrogen bonds, so they're really not very useful. Carbon dioxide is going to be like something like this, carbon dioxide. Is that it? Carbon dioxide. Yeah, I think that's the structure. And then H2O, as we know, is like so. And these bonds, right there and right there, they just don't have that much energy. But if we add light energy, energy from light allows the plant to do work called photosynthesis, which makes sugars. And sugars, the bonds in sugars have energy that living things can use more easily because many of those bonds are carbon and hydrogen bonds, carbon-hydrogen bonds. Okay, that energy later stored in chemical energy, maybe as a sugar, or as we've learned, maybe more like amylose or amylopectin as a starch in the plant, is stored and used to get work done later. Tissue growth, and all the functions of the plant. I won't go into it now. Okay, so chemical energy, that is the energy stored in bonds. Thermodynamics is the study of energy and its transformations, which sounds really cool. I wish I was a physics teacher now so I could say that I studied the energy and its transformations, but I'm just a biology teacher, so we get to study it for a little bit. We're going to look at living things for a moment just as a model, so as a closed system here, which means energetically speaking, living things don't interact with the environment, don't exchange energy with the surrounding environment. And then in situation B, this is seen as an open system because, and it's just a diagram, where energy can go in and energy can go out. So what do you think? Which diagram represents how organisms interact with their surroundings? A or B? Pause it if you want, but it's not that difficult. It is B. Living things are open systems and they interact with their environments where they gain and lose energy to the environment almost constantly. A couple examples of that. We are constantly losing heat to the environment, and we are pretty much constantly in need of taking in chemical energy. And the balance of how much chemical energy we take in, how much heat is lost, This is kind of generally connected to that topic of homeostasis, which we very briefly talked about in the past. Homeostasis, that means maintaining a stasis, homeostasis, which means maintaining a balance within the living organism between kind of an upper limit and a lower limit. For example, we don't want to get too hot, don't want to get too cold, don't want to have too much chemical energy, and don't want to have too little either. So we're going to be looking at how living things gain and lose energy in part, and the transformations of energy that are associated with that process. Thermodynamics. The first law of thermodynamics, which you need to remember, is number one, energy can be transferred or transformed, but cannot be created or destroyed. So what does that mean? It means that the energy in universe, what do you think? Is the energy in the universe constant, the same? Is it decreasing or is it increasing? It means it is increasing forever. It is for, what did I just say? That? Excuse me. It means that it is staying constant because energy can never be created nor destroyed. So within the universe, meaning all that is known, as far as you can go into space, uh, all of that stuff, the amount of energy is constant, but it changes form from chemical energy to heat energy to light energy, etc. This is one of the major themes in biology. Energy is transferred and transformed. So we talked about in chapter one, light energy or radiant energy can be converted to chemical energy. That'd be photosynthesis. Electrical energy can be converted to thermal energy. Chemical energy right here in the ice cream can be converted to mechanical or and then kinetic energy. And then chemical energy right here can be converted in the car to mechanical and then kinetic energy as well. So first law of thermodynamics, energy can be transferred or transformed, but cannot be created or destroyed. Second thing about that is in living systems, energy is always being transferred between organisms, such as between this plant here and the little critters that are eating it, the insects, and are all those insects? Yep, they're all insects eating this poor plant. And then the birds eating the insects. Energy is transferred, but it is not destroyed. And all of these living things are losing energy to the environment in the form of heat. Okay, the second law of thermodynamics is as follows. When energy is converted from one form to another to do work, some is converted to heat and is lost to the environment. One other thing that we can think about when we think about heat, I'm going to write heat right here. Heat is unusable and it is a chaotic. It is chaotic, it is random, and things in the universe, which we'll come to on the next slide, where we're going to talk about entropy in a moment, or maybe not quite the next slide. But anyway, heat is a very disorganized form of energy. So when we take something like A, whatever, and we convert it to B, and this B is sort of a more organized molecule, such as ATP. ATP. So we have an ATP is something that we really go bonkers for. So we'll draw some hearts for ATP. ATP is an organized molecule because it's a packet of energy that can be used to do more work later. It also took some work to make the ATP. So whenever we do something that we want to have happen, basically there's a cost, and that cost is a production of heat. You can think of it as any time that you want to say clean your room, well, it's going to cost energy. Heat is going to be produced. You did get some order in your world. Your room is now clean, or you've now produced some ATP. The cost is heat is also released, and heat is an unusable form of energy in terms of doing work. Okay, just couple other examples over here. Respiration takes this molecule, glucose, and turns it into a form of energy that we can call ATP. also some as heat. And when we do this, remember what's going on is we're getting some of that bond energy in these carbon and hydrogen, the bond between carbon and hydrogen, and we're using some of it to get something done that we want, ATP production. And the cost is heat, which is lost to the environment. Same thing with the whole world. Every time the world is doing something like photosynthesis or respiration, it is also producing heat, and that heat is lost to space in the form of radiation, like infrared radiation. That's why we cool down at night. Light shines away from the Earth in the form of infrared radiation. Over here to the right, in this diagram, what we can see is a whole bunch of energy transformation. So sun's energy gets used by these plants right here, the plants or producers to do photosynthesis. And when photosynthesis occurs, well, some energy is lost as heat. Plants also do respiration, so more energy is lost as heat. About 90% of the energy that plants ever get from the sun is eventually lost as heat. That's a bit of a simplification, by the way. And then when a consumer, such as a cow, comes along, eats primary producer, that energy is converted from one chemical form to another chemical form. And then these primary consumers, the cows, for example, are going to do work, such as they're going to walk around, they're going to perform all their life functions, reproduction, tissue repair, etc. And as a result, they lose energy to the environment as heat. So as you can see, if we have say if we had 100 kilojoules of energy with the primary producers, we're only going to have 10% of that left with the primary consumers. So I guess that'd be, well, 10 kilojoules here. Some of that's lost as heat. Most of it's lost as heat, about 90%. So when we get to the secondary consumers, so that would be predators, so maybe a wolf that eats the cow, then we only have 1% of our total initial energy. So we're down to 1 kilojoule. And this is one of the reasons that you generally don't see cougars and wolves and big predators on your way to work. There's simply not as much energy available to them because at the lower energy levels, all the way down here with the plants and consumers, that energy, when it gets transformed from each level to another level, energy is lost. And also within each energy level, the primary producers, for example, they use most of the energy. A little bit gets to the second primary consumers, the second level here, but then again, whatever energy they get, they use most of it to carry out their life. And so that's why those top predators, we don't see a whole lot of them. There's a lot less energy available to them because they've got to get it ultimately from plants, and because of their life strategy, they eat things that also don't have much access to energy. So entropy, this is one of the big ideas of this chapter. Entropy is a measure of disorder or randomness, and in general, a general rule of the universe is that all systems tend towards disorder, which we all know looking at this diagram to the right. Here, more entropy. Here, less entropy. Why is room A, why does it have less entropy? Well, because somebody spent energy to do work to clean it up. As a result, heat was emitted from their body. That's definitely a big simplification. Over in room B, the person did not use energy to do the work of cleaning up their room, so therefore it is a more chaotic space. And this simply happens naturally over time. Things do not, you don't randomly set things down in the correct place, just as a child doesn't. If a hurricane came through the room, it is going to tend towards more randomness, more disorder. Just by chance, if a hurricane comes through the room and blows everything around, right? By chance, things are not going to get blown into the right place. Now, is there 1 in a billion, zillion, trillion chance that that does happen? Sure. And it's a really, really small chance. That's where we'd use that whole thing, scientific notation, because the chances are so tiny, it basically is never going to happen. So when things happen in the universe, and by things I mean anything, the result tends to be chaotic because the results are generally random. And so what? So that is what entropy is. Okay, so entropy increases in the universe every day. For example, this mouse is losing heat. Heat is a very unusable form of energy, and that is an increase in entropy. Heat equals entropy. We could say that heat has a high entropy value. Let's see. The mouse is also excreting carbon dioxide, which is now a diffuse molecule in its environment. That is, we could call that an increase in entropy. And to prevent its internal environment from following that pattern, that is, down the road of entropy, chaos, and randomness, which means death, the mouse has to take in energy to do the work of being alive. And when it takes in that energy to do the work to keep itself alive, heat is produced according to the second law of thermodynamics. So a couple of notes to myself, because this is more of a physics topic, so sometimes I add some notes to myself to make sure I didn't forget anything. So energy is converted from one form to another. So for example, when the bread is eaten and some of it is lost as heat, this is an increase in entropy. And also the mouse used some of the energy in the bread to do work, the kinds of work that keep it alive, like active transport, uh, cell replacement, tissue repair, etc. Entropy in an organism can decrease, so that means less disorder at the cost of an increase of entropy to the rest of the universe. So as I said, all things in the universe tend towards an increase in entropy. And what the grandest scale of that is if you look at a picture of the stars and the universe and the galaxies and all that, well, everything and they kind of go back and forth on it, but basically they think it's all sort of spreading out, but maybe not as fast as it should be, but it seems to be spreading out. And it seems that the universe just sort of tends towards chaos, towards a spreading out of everything. So for the mouse to prevent itself from spreading out into decomposed materials such as CO2, heat, and other chemicals, it can go against this law of the universe. That is, that things get more chaotic. It can go against that law by spending energy, spending energy to decrease entropy in itself as long as there is an increase in entropy outside of itself. And that increase in entropy is heat. So overall, when the mouse is eating this bread, it is using some of that energy to order the molecules inside of itself, but it's a law of the universe. There's still an increase in total randomness because the mouse takes this relatively ordered set of molecules, the bread, they're all lined up in happy little starch granules. It takes these ordered packets of energy, energy, and uses some of it to order the molecules in itself, but it produces a lot of heat and increases chaos in the rest of the universe. So it uses the energy to repair damaged tissue, fight infections, excrete waste products, catabolic and anabolic pathways. How we can remember which one is which, they are opposites of each other. One takes small molecules, makes them into bigger molecules, and the other one takes big molecules and makes them into small molecules. So catabolic, this one's the one that I use to remember the whole thing. Catabolic, the root word here is kata. You can think of catastrophe. So catastrophe, yeah, we're not building anything in a catastrophe. Catastrophes are where things fall apart and break down. So that is catabolic pathways are where we take big molecules like carbs, fats, and proteins, make them into small molecules like water, carbon dioxide, and ammonia. This is called catabolism. So metabolism has two general categories, catabolism and anabolism. So the second one over here, anabolic. Anabolic is when we take down here, start with these small molecules, small, right? You can see that. Precursor molecules, small precursor molecules like amino acids, monosaccharides, etc. And we're going to make them into bigger molecules like the proteins we talked about, polysaccharides, lipids, and then we haven't talked much about DNA yet. Come back to that. So, how do you remember anabolic steroids? Well, you can remember catabolic catastrophe. That's a good way, and then anabolic's the opposite. You might also remember anabolic steroids if you're a sports fan or if you watch the news. Athletes are often getting in trouble for taking anabolic steroids. Anabolic steroids push the body's general metabolism towards anabolism. Anabolism means that we are going to make bigger molecules like bigger muscles, bigger bones. Those things are helpful if you're a competitive athlete. Okay, so let's see. Let's start back over here and talk about energy yielding nutrients. They are broken down by catabolism. We'll talk about what oxidative means later, and exergonic. Essentially, exergonic means it releases energy, but I don't want to get too into that. What is going on here is we are breaking down these big energy yielding nutrients into small, essentially waste products, and we're taking some of the bond energy, and again, generally the bonds that we're most interested in are the bonds that connect carbon and hydrogen. And we're taking this bond energy and we're transferring it to a useful molecule called ATP, also NADH, but we haven't talked about it yet. So we're getting some work done. So this production of ATP is, we have accomplished work. And why do we say that we have accomplished work? Well, work is something useful. ATP, what is ATP? Well, ATP is adenosine triphosphate, whatever this adenosine is, we'll just call it adeno for now. And then connected to it is 1, 2, 3 phosphates. And this is a decrease in entropy. Why would we call it a decrease? Because we've just ordered these molecules up into a line, like they're literally lined up. So what that means is if we have a decrease in entropy here, then we must have had an increase in entropy somewhere else. Where? Well, we lost a whole bunch of heat energy, and that was used to do work of producing ATP. ATP is also a molecule that can do more work later. And again, that is also when the energy in ATP is used to do work, it's going to lose energy as heat. And what we can do is we can take ATP and use it to produce things like these big molecules, proteins, polysaccharides, lipids, DNA, or nucleic acids. And we're using the energy in ATP to build things that we want. And all of this requires energy. That's why when you think about yourself eating plants, think of us as if we were all vegetarians and we would be the primary consumers. And to take all of these carbs, fats, and proteins in plants and convert them into us, meaning people proteins, people polysaccharides, fats, and DNA, we lose a huge amount of energy because first we have to break them down into smaller molecules to get this ATP, and then we have to take the energy in ATP and transfer it to all these small molecules that we have obtained in some cases from our food, and we have to convert them into what we want them to be. So it's not very efficient, and a huge amount of energy is lost. Okay, so now one of the maybe more difficult topics of this chemistry physics subunit in this chapter, free energy. Boy, free energy. If you are above the age of maybe 29, then you would like to have access to the seemingly free energy that people below the age of 29 apparently have. Free energy. What on earth does that mean? A crazy idea. Well, definition is right here, the amount of energy available to do work in a biochemical reaction. So unfortunately, it's not like the willingness to go out and have fun, no, or and how much energy you have for it. It's doing work in a biochemical reaction. So bio means living and chemical means molecule. So how much energy is there available to do work in a given biochemical reaction. Here is our equation. And free energy is G right here. That is what the free energy is. So free energy equals, don't get intimidated, you're not really going to have to do math here. You're just going to have to do simple math such as, is H bigger than T times S? Yes or no? And you'll see, it's pretty simple. So free energy, G, is calculated as, it's equal to H. What is H? That is enthalpy. And I encourage you to actually write this equation down and label each of the variables as we go. It is, free energy is equal to enthalpy, which is a complicated word, but it's really simple. It's the amount of bond energy present. It's the amount of bond energy present, like in glucose. So great. However much bond energy we have, that is the amount of free energy. That would be true in a perfect world, but the world is not perfect, and you cannot harvest all of the bond energy to get work done. You have to subtract something. So what takes away from the amount of bond energy? Why can't we take all the energy in the bonds and use it to do work? Entropy. This is the S. That's just S. S. Entropy. That is the amount of disorder, the amount of randomness. So molecules are small, crazy, fast-moving little fellas, and because of that, we cannot take them, have them line up in a nice, neat little row, and have them each donate their little packet of energy so that we get all of the free energy. It doesn't work that way. So randomness takes away from the total amount of free energy available. So let's start again with G. I'm going to change colors here. G equals is free energy, and it's the amount of energy in the bonds minus however chaotic the system is. Minus however chaotic the system is. The last one is T, temperature, in kelvins, and you have to use kelvins because this would be a different number if you converted it to degrees Celsius or Fahrenheit. So it's got to be in kelvins. Kelvins is a really irritating one. We don't really need to get into it though, because we're not going to be doing a bunch of math with it. Okay, so bond energy, we should be able to use it as free energy. Let's just use it to get work done in the cell, except not even close. The bond energy is not all available because the more temperature is and the more disorder there is, the less of that bond energy is available, which can be transferred to a given system to get work done. So temperature, let me ignore that for now. I got a little ahead of myself. Temperature, as temperature increases, what we can see here is that G is going to decrease. So as temperature increases, G will decrease. And that's simply because there's a subtraction symbol right here, right? So G is equal to H minus T. Forget about the S for now. So H minus T. So the bigger T is, the less energy is available. Now think about why that might be. Well, the higher the temperature, that means the more these little molecules are going in all different directions, and it is also the faster that they are going in all different directions. So that is sort of related to this entropy idea, meaning chaos and disorder. So we can kind of fuse these two concepts, especially for this class, because we really just need to understand it in an abstract sense. The hotter it is, and the more of whatever this S thing is, the more disorder there is. Well, just same thing again, more disorder. Hotter means more movement of molecules, and then S is already disorder. So the more disorder there is, the less of the energy in bonds there is available that can be transferred and used as free energy. Okay, I'm going to erase all this stuff so we're not too chaotic here. Okay, so some example values though. So right here, if S, or entropy or disorder, is 0, what that means is that there is perfect order and all the bond energy is zero, and all the bond energy is available. So why is that? Because if S is 0, T times 0 is 0, right? So this whole part of the equation is gone. It's zeroed out. T times 0 is just 0. So that means that all the bond energy is available to do work, but that is never possible, never happens in real life because the world is a chaotic place. As S increases, less of the bond energy is available for use in living things. So as randomness increases, less of the bond energy is available in living things. If T increases, can you explain why there is less energy available? You can pause the video and try to put that in your own words. I already sort of answered it. See if you can put that into your own words out loud, or if you're in a library, don't do that out loud, or if you don't want to look strange as if you're talking to yourself or your computer, maybe don't do that. But think about it. If T increases, can you explain why there's less energy available? You can pause the video and give that a shot. Okay, so free energy and delta G. Delta is simply something that means change. Triangle equals change. So for a given system, free energy and delta G. For a given system, like a cell or a chemical reaction, so whatever system we define, delta, this symbol shows the change between the beginning and the end of a reaction. So for example, delta G equals G sub F minus G sub I. Sounds really complicated? No, not really. What does G sub F mean? That just means the amount of free energy in the final products. And G sub I means the amount of energy in the initial product. So we could, if we wanted to make this a little simpler, we can't because this is simply not how the formula is written. Final minus initial. Yeah, that is what this whole equation is essentially saying. Change in the amount of free energy is equal to the amount in the final system minus the amount in the initial system. Next, we've got to define another term, and that is delta H. Delta H, that is the amount of bond energy in the final products minus the amount of bond energy in the initial products. Don't get intimidated. We'll go through it slowly. Starting here. The change in available energy equals the change in the amount of bond energy. So we're going to ignore this for now. This should be pretty understandable. Change in the amount of free energy equals the change in the amount of bond energy. And remember, that's in a simplified world where bond energy can simply be used as free energy. So if you use some of the free energy up, i.e. the initial amount in the bond energy, then you have used up some of the initial amount of free energy. So therefore, the final amount of bond energy is going to be similar to the final amount of free energy. So therefore, delta G equals delta H. Okay, unfortunately, we also have to think about the rest of the equation. Temperature, and you can think of this as the hotter it is, the more molecules are moving around, running around like a bunch of fifth graders at recess. And then also, so the higher the temperature, the more difficult this whole thing is to control in a productive manner. Delta S, that means the change in entropy. Okay, so this is where things get kind of freaky. A change in entropy, that means a change in disorder. So what do we think? As reactions go, this is sort of looking at a reaction because we're looking at the initial reactants and the final reactants, and the initial free energy and the final free energy. So it's like a chemical reaction. What do we think is that when we go through a chemical reaction, are we generally going to have more disorder at the end or less disorder at the end? Well, the answer to that is that we are always, in terms of the universe, going to have more disorder at the end. That's when reactions will spontaneously happen. Because why? We don't know, but it's a law of the universe. Could say a law of physics that reactions that produce chaos spontaneously happen, like your room getting dirty if you don't put work into it. So usually this is going to be a positive value. Now looking at an example for this equation, delta G equals delta H minus chaos and temperature. For example, if we were looking at the aerobic respiration of glucose, what happens is the bond energy, that is bond energy available in generally between carbons and hydrogen, is used, and that's sort of free energy that is related to free energy, except that of course chaos and temperature are also related to free energy. The products are CO2, H2O, ATP, and heat. Heat can generally be seen as an increase in entropy, and ATP is actually more of a decrease in entropy because ATP is a molecule we want. ATP is order. ATP is some kind of, it's called adenosine triphosphate. If this is adenosine, and then it's going to be connected to those three phosphates here. And this is, as you can see, pretty ordered. 1, 2, 3. So that's more order. So this is a decrease in entropy, production of ATP, and then the release of heat is an increase. Okay, let's look at some math. Let's say T equals 1, because 1 is really easy for me to multiply. And let's also say delta G in this reaction leads to less free energy available. The amount of bond energy, also less bond energy available at the end than there was at the beginning. And to make the numbers work here, if there is, excuse me, after this reaction has occurred, negative 10 kilojoules of free energy change, so there's less 10 kilojoules of free energy. And if I just made up a number and said there's a less 5 kilojoules of bond energy, then that would mean that the change in entropy would have to be 5 to make this equation work, because negative 5 minus 5 is negative 10. Negative 5 minus 5 is 10, especially, temperature is 1, like I said. So what can we say here again? Firstly, free energy cannot be directly measured, so it's not really a hard science here exactly. But we can say that there is less free energy available, so less energy available to do work. There's about half as much bond energy as we have lost about half of that in bond energy, and we have had an increase in disorder and randomness. Remember also these deltas, these triangles, what that essentially means is delta S would be S sub final minus F sub initial. So if T is 1, we can just ignore it, right? So if I was going to do the math to make delta S equal 5 here, what that would be is, let's say I get a better color. I'll get black. So S sub I here, let's say that there's perfect order in this magical example, then the final is going to have to be 5. 5 minus 0 is 5. So hey, wow, aren't I a math whiz? So we get a delta S of 5 there, and a positive value for S means more disorder. Positive value is more disorder. So again, what happened here? There's a positive value for disorder. We have lost some bond energy because some of that bond energy went to probably increasing the disorder in the universe, as we can think of as heat loss, heat. And there's this much less energy available to do work. Exergonic reactions. They are reactions where the reactants have much more energy than the products. Typically, the difference in energy is the amount of heat that has been lost. For example, respiration. Respiration takes food molecules, food like C6H12O6, and produces some type of product like ATP, CO2, H2O, and then of course also loses a bunch of heat. The only useful thing here is ATP, and CO2 and H2O are essentially waste products. The amount of free energy available is less because these products have less energy than the reactants. So again, initial could be, I don't know, we'll say 20 kilojoules of energy were available initially. And then final amount of energy available, we'll say, oops, we already know there's a G there. Final amount of energy available there, we'll say was a 5. So the delta G here is going to be 5 minus 20, which is negative 15. We lost negative 15 kilojoules of free energy, but hey, we did get some ATP, which is a useful molecule, as we shall see later on in the chapter in the course. Endergonic reactions, excuse me, endergonic reactions take in energy. So a good example would be sort of the opposite of respiration, which would be maybe photosynthesis. Photosynthesis, which is CO2 plus H2O. These things are not food molecules if you haven't noticed. You can't really get energy from water, and you certainly can't get it from CO2. But if you add some energy from, say, sunlight, and you have a plant to do all these really complicated chemical reactions, you'll get a product like C6H12O6. That is a food molecule. You can eat it. It has a lot of energy. So again, that delta G now, delta G equals G final minus G initial. The initial products, initial reactants are going to have very little energy. We'll say 5 kilojoules. And the final reactions have a lot, say 20 kilojoules. I'm making these numbers up. 20 kilojoules. 20 minus 5 is 15. Positive value for delta G. There's more free energy available to do work. Okay, yeah, and here's a graph of it. Reactants, course of the reaction, products have more energy. Where did the energy come from? From some outside source of this system. If it's a plant cell, then obviously it would have come from the sun. Diffusion is an exergon process. That means that it is an energy releasing process. Here it is. Concentration gradient, diffusion, is going to go that way. This can also be seen as free energy. Therefore, it can be used to get work done. Like water flowing down down the river, we can harness it by putting something in place like a dam or like a rotary thing that, when something goes through it, it spins and you can make electricity. And guess what? Cells do this by creating these concentration gradients, except they're not funny little balls. They're protons or positively charged hydrogen ions. We put them all on one side of a membrane. That represents free energy. Cool. Why is it free? Well, I mean, nothing's really free, but we can call it free. They'll diffuse down their concentration gradient, and if the cell has installed one of these fellows here, this thing, it's a huge protein complex, and it'll actually rotate, and in the process of rotating, it produces ATP. So we are harnessing free energy here in the form of a concentration gradient to do work. What is that work? To produce ATP. The process happens spontaneously because exergonic processes are like that. They're energy releasing, like, say, lighting gasoline on fire. It doesn't require a whole lot of encouraging. And in this case, the diffusion example, it happens until equilibrium is reached, as we know. Let's see here. After it's all said and done, we have greater entropy. Here, greater entropy, because there's disorders just kind of everywhere. We haven't said, hey, all you fellas, be on the left, and we don't want anyone on the right. That's how we order things in this world. Here, disorder. And yeah, I said it all. Next, free energy changes depend on the concentrations of reactants and products. Oh dear. They depend on the concentrations of reactants and products. Let's take an example. CO2 will spontaneously form carbonic acid. Equally and reversely, carbonic acid will spontaneously form carbon dioxide. So these equations, they go back and forth. They go back and forth. As you can see by this symbol, they can go back and forth. So if there's lots of CO2, then CO2 breaks down into carbonic acid. This is a spontaneous reaction. It has to do with how these bonds are more and less stable. It's kind of like somebody sleeping at night. They toss and turn. I think I want to be in my CO2 position. And then later they, now I think I'm going to be in my carbonic acid position. So from one to the other. If you've been in the CO2 position a while, you're thinking, I think I'll try carbonic acid. Equally and oppositely, if there is a lot of carbonic acid, then it is going to spontaneously convert itself to CO2 because it's just not happy sleeping all night in the form of carbonic acid. It's not stable. It's not perfectly stable. It goes back and forth, back and forth. If there's more carbonic acid, then randomly just have more of the reactions going in that direction. If we have more CO2, then randomly we have more of these reactions going in this direction. Okay, real quickly, the arrow shows that it's reversible. This is called dynamic equilibrium. They flip-flop back and forth, kind of like you sleeping, or at least me sleeping, I'll tell you that. CO2 and carbonic acid, dynamic equilibrium. Reduce the concentration of one molecule and the reaction goes in that direction. Or, so if you made more CO2, then it'll go to the right, forming more carbonic acid. Okay. Now, let's take a look at what Lisa Simpson is doing and what I always wondered about in movies where the panicked person starts breathing into a paper bag, and I'm thinking, no, don't do that. There's no oxygen in there. You're going to run out of oxygen and suffocate. You must be crazy. Nope, you're not crazy. It doesn't make sense. So, in the non-panicked situation, cells of the body, they are doing aerobic respiration, producing CO2. In the blood's just continuously getting more and more CO2 from all the cells in your body that are respiring, so we get more and more carbonic acid. And long story short, because this is not anatomy and physiology, but there's a certain amount of carbonic acid that the blood wants to have because pH is very important to balance in the blood. Now, so keeping that in mind, there's a certain amount of carbonic acid that is supposed to be in the blood, and that's good. Now, what about Lisa Simpson here on the right? We're going to say that she has panicked, and now she's breathing in and out of a paper bag over and over again. So what's happening is she's breathing out. Oh, sorry. Let me rewind a little bit. Before she had the paper bag, she's panicked, right? And so she's hyperventilating, going like, that probably sounds weird in the recording, but if you breathe in and out really rapidly, what's happening is you're exhaling too much CO2, loss of too much CO2, which means carbonic acid is spontaneously converted to CO2. So there's less carbonic acid. There's less acid in the blood, and if you lose too much acid in the blood, that disturbs the pH balance of the blood, and you can lose consciousness. So how can you just make your blood more acidic if you want to? Well, and I am not a doctor. Here's my disclaimer here. I am not a doctor. Do not try this at home. But what happens in cartoons is the person is hyperventilating, so they need to increase CO2 or carbonic acid in the blood, so they breathe into the bag and they exhale CO2. Then they inhale that same CO2 at a higher concentration because there's basically no CO2 in the air. It's way less than 1%. So exhales it, breathes it back in. Now she exhales even more CO2. Now she inhales even more CO2. CO2 is building up in the bag. So when you inhale lots of CO2 from the bag, you are increasing the amount of CO2 in the blood also because it diffuses back into the blood. And you're pushing this reaction back in the carbonic acid direction, replacing that lost carbonic acid and coming back to a state of uh, homeostasis in terms of blood pH. Which of the following reactions will happen spontaneously? Your bedroom cleans itself, or your bedroom makes itself dirty somehow? Well, I'd like to believe that my bedroom will clean itself. My wife would also like to hope that that would happen. It won't happen. Well, it's our bedroom. Okay, enough about me though. One, in the world of chemistry, A to B delta G is a positive value. So great, in this reaction, we just have more free energy because it's a positive value for G, delta G. So is that how life works? You just have more free energy? Well, last time I checked my electric bill, that didn't happen. Number two, C is converted to D, and there's a loss of free energy. That seems more, well, maybe pessimistic, but also sadly true. Yeah, this is the one that'll happen spontaneously. This is the one that's more like, I lit a tank of gas on fire. Kaboom! Now there's a lot less free energy. Also, don't try that one at home either. That's very dangerous and you will be injured. But negative values for delta G happen spontaneously. And again, the real world analogy is just an analogy. It's like money. Does money spontaneously accumulate in your bank? No, it kind of spontaneously disappears. I was checking a credit card balance the other day. I was like, how, it can't be. This can't be true. It seems to spontaneously happen. And it's always in the negative direction. That's how the universe works. However, we can couple reactions in the, I'll leave the analogies for now. Some of you will be thankful, I'm sure. Coupling reactions, so the A to B one is not going to happen spontaneously because there's more free energy available at the end of it. C to D will happen spontaneously because there's a negative delta G. You want to memorize that. If delta G is negative, it spontaneously happens. If we couple them, we join them together, then we get, let's see, positive 20.9 plus negative 33.5 is negative 12.6. So basically, the energy yielding reaction here can be coupled with the energy consuming reaction here, and thus the reaction happens. Kind of like me coupling work with my endless expenditures. So what happens is A plus C forms some, we'll just say intermediate product, and that's a negative value because, well, why? We'll ask a chemist. But basically we've got this C to D component of the reaction, and that yields free energy. So C is eventually getting converted to D. So A to C, though, we're coupling the reactions. A to C forms an intermediate product. Intermediate product forms the same products. And the formation cumulatively of these two products from these two reactants, cumulatively, the delta G is negative. Therefore, the reaction will occur. This is a math model. Here is a biological model. Active transport of we're going to call this glucose up its concentration gradient. What does that do? This is going to have a delta G that is greater than 0. It's a positive value because we're creating order in the little universe of this cell by creating a concentration gradient. We're ordering things. You're saying all the glucose molecules must be on this side of the membrane for some reason. Okay. So get over there. We're ordering things. So that means that we're going to have a loss of free energy. We're going to couple it to, we're going to couple this reaction that has a positive delta G value to a reaction that has a negative delta G value. I think this one is negative delta G. When we phosphorylate a mole of ATP to produce ADP, I believe the free energy is, I think it's 32 kilojoules per mole. I might have forgotten the exact number, but something like that. Lots of free energy available in this reaction. So this reaction, we can couple it to the transport of glucose up its concentration gradient. Why? Because it releases energy. And this bond here specifically has a lot of energy in it. And this energy can be harvested by adding it to, over here, come on, follow what I'm talking about, over here, adding it to this protein transport pump. And when we add this phosphate to it, it undergoes a conformational shape change, pumping the glucose up its concentration gradient. And this reaction sorry, I should have said negative. Didn't I say negative? Negative. Has a very negative delta G. So it'll happen very spontaneously, and we can couple it to reactions that are useful to the cell. See that? Useful. Wait, is useful with two L's? I'm tired. I didn't have enough coffee this morning. Hydrolysis of ATP is exergonic and is often coupled to endergonic reactions. So ATP is hydrolyzed. Oh yeah, I remembered it correctly. It's got a negative 32 kilojoules per mole change in free energy. So lots of energy is released to the environment as heat. Some of it we use to do useful things. Example, we are going to take this reaction, glucose plus fructose, we make it into a disaccharide. Each of these are monosaccharides, if you remember. If we want to make it into a disaccharide, say for example we're a plant, plants have to transport all of their sugar in the form of sucrose for some reason. And so they've got to take anything that they want to move around, glucose and fructose, turn it into glucose. Problem is, it's got a positive delta G. That means sucrose has more energy than these two molecules. So it won't happen spontaneously. We couple it with the above reaction, the hydrolysis of ATP. By doing this, we are going to hydrolyze ATP and phosphorylate glucose. This is a high energy bond. It came from this negative delta G here, the hydrolysis of ATP. And this high energy bond basically represents chemical potential energy that has been added to glucose. Glucose can now react spontaneously with fructose to produce sucrose. You do not have to memorize this equation. You have to understand that energy yielding reactions can be coupled with energy requiring reactions to make these energy requiring reactions proceed. I could, however, show you all this information again and ask you to comment on it in some way, but you don't need to worry about memorizing these values or the reaction. ATP for cellular energy. One more time. If the first one or two times wasn't fun enough, hopefully this one will be fun. Exergonic reactions, these things release energy. One of those exergonic reactions is where we take our food, yep, pizza. The whole reason you eat it is so that you can make ATP. Cells sound like they have a boring life, don't they? Well, ATP this is a high energy molecule. Now, you might be getting a little confused here thinking, Mr. McLeod, you said this is an exergonic reaction. It's releasing energy. And now you're saying ATP has a lot of energy. Well, some of the energy was put into this useful form. Much of it was lost as heat. Put your hand on your head. You feel heat? Most of that heat is coming. Yeah, most of that heat is coming from this reaction, taking the food, making it into ATP. ATP can store the molecule. It's everywhere in cells by the billion, zillion, trillions, and it can be coupled to endergonic reactions here that require energy, like active transport, building new cells, transporting vesicles around the cell, and ATP is hydrolyzed, producing ADP, an inorganic phosphate, which can be reused by adding some food energy to it, and then we get more ATP again. So this is sort of a cycle. Oh boy. Over here, to make absolutely certain, you know what is going on when we hydrolyze ATP. It is being hydrolyzed just like the hydrolysis of a polysaccharide or a lipid or a protein. Water is being added to this bond right here, and that process leads to the release of a large amount of energy. Two hydrogens and one oxygen. Notice in this bond here, there's just one oxygen. Down here, there's a second oxygen and one hydrogen that's been added here, and then there's a hydrogen that's been added here, and then here's our original oxygen. So this is, again, also, you don't need to memorize where all these atoms are going, but you do need to know that ATP is being hydrolyzed using water to split that last bond, like that. Atomic structure overview. Number one, this one has got to be hydrogen because it's got one proton and one electron. Yes, I got it right. Second one has got to be, let's see here, I'm going to count the electrons. We have 1, 2, that's how many fit on the first energy level. And then on the next one, we've got 1, 2, 3, 4, 5, 6. Okay, so it must be oxygen. Yes, right again. Oxygen wants to fill this outer energy level, wants 8. So we say that oxygen is electro negative. That means it's really attracted to electrons. It wants to gain 1 and 2. So it could form 2 covalent bonds, say with hydrogen, forming this magical molecule we call water, except I forgot how to draw it. Try that again. This magical molecule called water. See, hydrogen, hydrogen, oxygen. All right. Over here, this is another atom, and it's got 1, 2 electrons on the inner shell, and then 1, 2, 3, 4 on the outer. Must be carbon. It can form 1 bond here by sharing an electron with something else. Another one here, another one here, and another one here. It could form, in fact, 4 bonds with hydrogen. So if we took a hydrogen here, hydrogen's 1 electron would hang out right there. That would be a covalent bond. And then the same thing would be possible here. So hydrogen, and then etc. Like again, etc. Right, etc. So 4 of them, and then that would form methane. If you ever were curious about that. Okay, I digress. Inside the nucleus are protons. They're positively charged. There are also neutrons. Atoms can occasionally lose electrons. They can also occasionally gain electrons. And that's what we're going to be mostly interested in now. Oxidation reduction reactions. So oxidation reduction, that's where we get the name redox reaction. So re for reduction, ox for oxidation. And for now, we're going to focus on just one part of what you need to know, and that is, are electrons being gained or lost? So here's how you remember it, and I promise you it'll make it a lot easier if you actually do remember this. Leo Grr. Leo, the lion says, grr, Leo grr. So losing, L stands for losing electron, and then E stands for electron. Sorry, my writing with my mouse isn't very good. Is oxidation. Okay, so now let's look at an example over here. Hello, right here, that one. This one, see, it's got an electron right there, and we're just saying it general blob, whatever it is. It's got an electron, and as this reaction progresses, here's the reaction arrow, then this fella has lost an electron, huh? Where'd it go? It's gone. So it has been oxidized. Leo Gur. Lose electrons, we say it's been oxidized. Gur, Leo the lion says, Gur stands for gain. E again stands for electrons, and R stands for reduction. Reduction or reduced, both the same thing. So this fella is the electron acceptor, and it's been reduced because it has gained an electron. Gained an electron, it's reduction. Okay, over here, same thing. A is going to be oxidized in this reaction. B is going to be reduced. What's the point of showing this one again? Well, that's just another cartoon, and I love cartoons. Redox reactions are paired together pretty much always. So, and why is that? It's kind of like, a business transaction. Someone gives money, other person accepts money. So A is going to be oxidized, B is going to be reduced. A gave the electrons to B. So B, think of it, Leo-ger. B gained electrons, so it was reduced. A lost electrons, so it was oxidized. Here is what you actually need to know about redox reactions. A little bit more. So oxidation is on this side, and it is not only the loss of electrons, it is also the loss of hydrogen or the gaining of oxygen. Oh, that's so annoying. Here's how I want you to think about it. Well, it's not how you have to think about it, but it's how I think of it. Oxygen is like that person who you invite to your pizza party and they show up early and eat all the pizza. Oxygen is a pizza pig, okay? Except we're obviously not talking about pizza. Oxygen is electronegative, so oxygen really likes electrons, and it'll also really, well, we're just going to say it really likes electrons. So think about it this way. Oxidation, if you gain an oxygen, it's like a loss. Why? Because oxygen is going to show up early to your party and eat all of your electrons. So gaining an oxygen is like losing electrons. Kind of like gaining that friend is kind of like losing pizza because they show up early and they eat all your pizza. I hope you found that useful. Reduction. Reduction, let's go back to oxygen here. If you lose oxygen, it's like gaining electrons because it didn't eat all the pizza, right? It didn't suck all the electrons to itself. So reduction, we consider that to be a gain of electrons or a gain of hydrogens. We'll come back to this time and time again in this chapter and also the next. Oxidation reduction reactions, again, redox reactions. Let's look at an example for the production of ATP. So, glucose has lost a hydrogen and the electron attached to the hydrogen. So, an electron and a proton, or we could also just say hydrogen, right? And it has been added to this molecule, NADH. NAD plus. NAD plus is an electron carrier. It's an electron carrier, and it is now reduced. How do I know it's been reduced? Because it has gained a hydrogen. Lipids, they have a ton of hydrogen carbon bonds, which is why they are so energy dense. They can donate lots of electron hydrogen pairs to NAD+, which will carry them through a series of reactions producing ATP. So ATP can then be used to do work. That is why fats, we say, have a tremendous amount of energy. All those hydrogen carbon bonds can easily be broken. And they are high energy and they are used to make ATP. Use that for active transport, anabolic reactions, DNA, protein synthesis. Finally, can you think to yourself, and you can pause the video, which molecules are oxidized and which are reduced, and also which reactions are endergonic, which are exergonic. I'll add a few comments. You can pause it now. Okay. Glucose and fats, or the lipids, those were oxidized. NAD was reduced. ATP, this is a little bit more chemistry. ADP plus inorganic phosphate. This was reduced to become ATP, but that's a little more chemistry than we need, so don't worry about it if that's confusing. NAD was reduced, I mentioned that. Which reactions are endergonic, which are exergonic? Well, this whole sort of thing, this is, and then also from glucose to produce ATP. If you remember, the production of ATP from food, food molecules is exergonic, meaning we're also going to release a lot of heat. More details on this molecule, NAD+. So we can see, here's the structure of it. You don't need to memorize this structure. But what I do want to convey to you is that this molecule, NAD+, and then it reduced to NADH, you should have a little bit of understanding that there are bonds being created here. NAD is actually more closely related to like DNA or RNA than it is to a protein or anything else. It is called nicotinamide adenine dinucleotide. The whole thing isn't written here, but you don't need to memorize that, thankfully. NAD, nicotinamide adenine dinucleotide. It's because there's two nucleotides on here. Anyway, it carries these things: one H plus and 2 electrons. It gets them from food molecules, and it's going to add them right here. So there's 1 electron added to here, and then 1 H added there. And then the truth of the matter is that there's also an H plus added here, because 2 electrons, and there needs to be a pair, because remember, if we're taking these hydrogens, each hydrogen having 1 proton and 1 electron, they kind of come in a pair. Hydrogens always have those two things, and if we're taking them off of these fatty acids that have all of these hydrogen carbon bonds on them, where are the remaining hydrogens going? Well, in your book or online, you may sometimes see it written like this: NADH plus H plus. There is one extra proton that is carried here, not shown here. But it is carried in a dissociated state, meaning it is falling off of the molecule and then rejoining it, kind of like you tossing and turning in sleep. It's not stable in any one place. The level of when is it on, when is it off, and why is way beyond this course, though. But you just may see it written like this, so I would explain that. FAD and FADH2 is the exact same thing. It is also an electron carrier, and it carries things in a little bit different way. Firstly, you can see it is not charged over here. There is no plus, no plus, and it just gains 2 electrons with their 2 protons, so 2 H pluses, and they get bound right here and here. You don't need to memorize where they're bound, but you do need to know that FAD carries 2 hydrogens or 2 electrons and 2 H pluses, and they are bound to FAD. This will come back to us when we discuss aerobic respiration in the next chapter. Enzymes, new topic. Enzymes are like construction workers, except you don't need to pay them money. They take some materials and they make new materials, and they are not used up in the process, meaning the construction worker does not end up being part of the building. So as such, we call them a catalyst. They catalyze reactions, meaning not used up. They are also proteins. So that means we're going to review proteins real quick. Protein structure. There is primary protein structure, which is the sequence of amino acids, and that is programmed by our genes. Secondary structures. These are repeated things that occur repeatedly across many different proteins. We have alpha helices right there, and we have beta pleated sheets. These contribute to tertiary structure. Tertiary structure is when it acquires a 3-dimensional shape. Proteins, to my knowledge, are always at least proteins having 3D shape. Proteins always are going to have tertiary structure. Often are going to have a quaternary. I'm not going to write it out. It's written right here. Quaternary structure, where more than one of these polypeptides are joined together, as we've seen here. Here's one, and then here's the other, forming quaternary structure, the quaternary level of protein structure. They are here are globular proteins. Which one am I going to mention? We have already talked about hemoglobin, carries oxygen in the blood. Antibodies we talked about briefly. Antibodies are globular proteins. That's why they're called immunoglobulins, if you want a big word. This is alcohol dehydrogenase. If you take in alcohol, this is the enzyme your liver will produce to break it down. That's another component of alcohol tolerance, by the way. Some people naturally have more of it pre-existing, and also people can upregulate the production of this enzyme, so they'll break down alcohol more rapidly. It's also related to other things like receptor withdrawal as well. Over here, this is a big one for our next chapter, which is respiration. This is ATP synthase. And as I mentioned earlier in this program, what one form of free energy is a concentration gradient on one side of a membrane. So here, that's our membrane, the phospholipid bilayer here. And the cell manages to get all of the H pluses crowded into a tiny little room on this side of the membrane, and they are positively charged, so they repel each other, so they wind up diffusing through this whole apparatus. And this part here actually will rotate as the protons diffuse through, and as it rotates, it produces ATP. So point being, proteins are like big machines in many cases. So, for a reaction to happen, as I said, enzymes are catalysts. Catalysts. So they help reactions go. Let me take the example one more time of respiration, which is C6H12O6 and energy in the product. So this is an exergonic reaction. That'd be CO2 plus H2O and then plus ATP and heat energy, but that's sort of more than we want to get into for now. Alrighty then. Will this reaction happen spontaneously? Well, there's what did I say here? Delta G. I even have to remind myself sometimes. So the G final minus G initial looks like the G final is a little number, right? So that's smaller. And then initial, that's reactants, that's a big one. So the initial is going to be a big number, like say 10, and then the final products are, we'll say, a smaller number, like 5. So it's going to be a negative number, -5 in my simplistic example here. So what that means is this should occur spontaneously, but wait a minute, not really. If you've ever sat all day like I have and stared at a pile of glucose, it's a little powder. It's not a little powder, it's just powder. And I didn't see any carbon dioxide and water formed, and I definitely didn't feel any heat getting put off. Well, that's because it will spontaneously happen, but it won't spontaneously happen very easily. That's the thing. Why won't it spontaneously happen a lot, really rapidly, even though it's supposed to, because it's got a negative delta G? Because of this fella right here. Well, it's already shown here. Activation energy. Without an enzyme present, we'll forget the enzymes for now, there's quite a lot of activation energy. What could this come in the form of? Well, heat could increase the temperature, meaning these molecules of glucose are going to be moving around more, banging into more stuff. And that can provide the activation energy that will break this molecule apart into CO2 and water, but it requires a lot of activation energy, specifically raising the temperature. So if you've ever had a sugar cookie and then just decided that you want to digest it, so you lit yourself on fire to achieve the activation energy. Well, what that means is that you dramatically misunderstood the point that I'm trying to convey to you. That's another one of those don't do this at home things. You don't need to light yourself on fire to break down glucose. Why? Because inside of your cells, there are enzymes. And with the presence of an enzyme, the activation energy is lower. Activation energy with an enzyme, that's this slope here. So with an enzyme present, the reaction can proceed at a lower activation energy, which also often means a lower temperature. So it will proceed at a lower temperature. It will also proceed more rapidly. So enzymes, what we know about enzymes so far is they lower activation energy. I said they are catalysts. They also speed up reaction. So I was sitting at my kitchen table staring at the bowl of glucose all day. Maybe a couple of molecules turned into carbon dioxide and water I didn't notice, and I got bored and walked away. It was just too slow. But in your cells, this will happen really fast because there is a activation, because there are enzymes present. How does this happen? Well, an enzyme, there's the enzyme, will bind to its substrate. For example, the substrate could be glucose, as I just mentioned. Could be over here. We're saying substrate is hydrogen peroxide, H2O2, hydrogen peroxide. That's a toxin that shows up in basically all of our cells as a byproduct of some other reaction, and it's super toxic, so enzymes break it down. Anyway, glucose would bind to its enzyme, and it would bind to the active site. So substrate binds active site. On an exam, do not say that the substrate binds to the enzyme. Be specific. The substrate binds to the active site of the enzyme. Another thing, what about the structure of the substrate versus the structure of the active site? Do you notice? Well, you may be tempted to say they are the same shape. They're not. For example, if you put on a glove and it fits your hand, is it the same shape? No. What we say is they're not the same shape. We say that that the substrate is complementary to the active site, Just like a glove is complementary to your hand. Your hand is not hollow. The glove is hollow. The interior of the glove fits the exterior of your hand. Okay. Let's see here. So what happens is substrate binds the active site, and then the enzyme goes yum 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 yum, crunch, bites the substrate, and then products are formed magically. Great. A little bit more on this though. The induced fit model that we can see right here, this is the model that we are most interested in. In high school, you might remember the lock and key method model of enzyme catalyzed reactions. The induced fit model is a better, more accurate one. Why? Number one, the substrate is not, in fact, the same shape as the active site. In fact, it is not even quite complementary to it. So what I mean to say is that the substrate does not fit perfectly into the active site. So because it's not a perfect fit, kind of like putting on a shoe that is too small, the lack of a perfect fit stresses bonds in the substrate. And eventually, that stress provides the activation energy, lowers the activation energy, and the bonds are broken, and maybe new bonds will be formed with something else. Here we have enzymes, our biological catalysts continued, and we're going to look at what happens when a substrate, sucrose, which is a disaccharide, two monomers, they can be broken down into these two things, glucose and fructose, two monosaccharides. How does this happen? The substrate, sucrose, binds to the active site of the enzyme, probably going to be named sucrase or something like that. They usually name them after their substrate. The bond is not perfect, so this stresses the enzyme-substrate complex. That's a term you need to know. When the substrate here binds to the active site, this is called an enzyme-substrate complex, and this is what stresses the bonds. Like I said, stresses the bonds, lowers the activation energy. In this case, there's another substrate that is not shown really as a substrate, although it is. It's a reactant. H2O is involved in the hydrolysis of sucrose because water is split, and that breaks this glycosidic bond between glucose and fructose, forming the products, glucose and fructose. That is all I wanted to say on this slide. I already mentioned everything we need to know about that one. Cofactors and coenzymes. If you've ever wondered what it is that vitamins or minerals do, what they do is they often will make a substrate fit its active site better, or in some cases, the substrate may not fit its active site at all without a cofactor. So here, the reactive substance, the substrate, will not fit the active site. It's not complementary enough. But if we add in what was I saying? A vitamin or a cofactor. Co means like cooperate. It's going to be part of the thing, part of the reaction. We add this vitamin or mineral. It also binds to the active site, and now the substrate can fit into the active site as well in a way that catalyzes the reaction. This is and vitamins are present in basically all the vegetables and the fruits and the meats. More processed foods, you're going to get less vegetables. Enzyme catalyzed reactions in your body will happen at a slower rate, and that means that you're going to get less of what it is that your cells want. So eat your veggies. Things that affect how enzymes work. Specifically, factors that affect enzyme catalyzed reactions in terms of the rate of reaction. So we're going to now look at a bunch of graphs, and on the y-axis, that is the rate of the reaction or speed of the reaction. What this graph is showing is that as temperature on the x-axis increases, the rate of the reaction also increases until it gets to its optimum or maximum rate of reaction. We can also call this the optimum temperature. That's a word, phrase that you want to know that describes the peak speed or peak rate of reaction. Optimum temp. Optimum temperature. After this optimum temperature is passed, the rate of reaction begins to go down, and it goes down pretty rapidly, as you can see, until no reaction can be catalyzed. At this point, the enzyme has denatured, which we learned a little bit about in a past chapter. Enzyme has denatured, which means bonds have been broken in it. It has lost the conformation, the structure that enables it to catalyze reaction. So it has now been denatured. No reactions can be catalyzed. Let me make another note on this graph. This graph is not showing one enzyme catalyzed reaction as it progresses. The label on the x-axis here is not progress of reaction. So it's not as if they're slowly increasing the temperature until it goes really fast and then it slows down. This is not the progress of the reaction. It is the temperature of different reactions. So essentially, what is done to determine how temperature affects a given enzyme, there's thousands or millions or whatever, right, is you would have a whole bunch of test tubes at different temperatures, like, I'll use 5, 10, 15, 20, 25, etc., till we get to a really hot temperature. And they'd have one test tube that they're going to put at 5 degrees, another test tube at 10 degrees, another test tube at 15 degrees, and then they measure how much product has been formed, and then they graph it. Okay, so at 5 degrees it was this much product. Okay, we'll do the experiment again. At 10 degrees it was this much, and then at 15 degrees it was this much. And then they keep doing this until they essentially are able to plot a line graph showing the course of the reaction. So this data is the result of many experiments. Okay, over on the right side, ladies and gentlemen, over there, we have how the rate of reaction, again, speed of the reaction, is affected by two different temperature for two different enzymes. This one is a typical human enzyme right here, and typical human enzyme, and 37 degrees is body temperature. And what do you know? Body temperature appears to be the optimum temperature for most typical human enzymes. If you get a fever, enzyme catalyzed reactions start to drop off, and that can be a problem. I once had a fever of, I think it was 44, 44, right? And that is basically close to death. You run the risk of some other enzymes basically going off the charts and going really fast, actually. They produce a bunch of heat, and then it causes runaway, uncontrolled reactions, and the person dies. It was extremely unpleasant. I lost 1 pound every day for 5 days because my metabolic rate was so high. Here, this is the optimum temperature for an enzyme of a thermophilic, meaning a heat-tolerant bacteria. So this fella is really happy at about 80 degrees Celsius. 80 degrees Celsius, how much is that is in Fahrenheit? So let's see, what would that be? 80 plus 32, wait, is that how you do it again? It'd be about 180 or something like 190, something like that. Over here, the last thing we're going to look at on this slide is how pH affects enzyme catalyzed reactions in terms of their speed, in terms of their rate. Optimum pH for pepsin. This is an enzyme that is in the stomach. We're just going to forget this for now. Okay, this is a different enzyme. We're not interested. Optimal pH for this enzyme is very low. It is a pH of 2, and you've probably heard that, if you have enough of an acid that is a pH of 2, it'll break down nails given enough time, and that's how low the pH is of the human stomach. So enzymes that we produce that work in the human stomach have an optimum pH that suits their environment. 2. Why is it that some enzymes would be able to work in such a low pH and unfriendly environment? They would likely be fortified with a lot of things like disulfide bridges to fortify their internal and three-dimensional structure. Whereas this enzyme, now we're interested in this fellow here. Hello. I'm not interested in this one anymore. This is a different enzyme, number 2. And this is for an enzyme, trypsin, don't need to memorize that name, that occurs in the intestines. The intestines are comparatively alkaline, meaning they are the opposite of acidic, comparatively alkaline or basic. And enzymes that occur in this environment, the small intestine, are suited to that environment. They have an optimum pH of around 8, that is also the pH of the intestines. Denaturing proteins. So proteins have to be folded in a certain way. Part of that is protein modification, happens in the rough ER and the Golgi. When they are modified, they take on a specific shape that suits some function. If you have too much heat, then they can basically vibrate back and forth enough, because that's what heat is, causes molecules to move more, until they denature or unfold. This is a denatured protein. It is not reversible. Not reversible. Down here, this is how, so here I've kind of given you an example of how temperature can denature an enzyme. Here is how pH can also denature an enzyme. So what happens is, if we have too many fellas like protons, protons occur when we have acids, like in the stomach, and protons are positively charged. The problem with having a lot of positively charged things for whatever protein it is, fish muscles, for example, in this diagram, is that proteins, some of them have negative ions as amino acids. Some of the amino acids are negative ions, and these positives can react with them and cause the whole structure to change shape, thus denaturing the enzyme. On that last slide, I did not mention that, so I'm going to mention it now. If we have pH here, and this is the rate of reaction, so this is rate. What's happened here And what has happened here, quite likely, is denaturation. Denaturation. There were too many protons. If it was a low acidic environment here, that would be acid. And all these positively charged fellows interacted with the protein in such a way that it lost its structure. It could be a three-dimensional structure, whether it could be. Over here, this is a higher pH called alkaline, and that means that there's going to be a lot of these OH minuses. And all these OH minuses, they're also charged, as you can see, right? They've got a negative. So those negatives can interact with positive parts of amino acids and proteins and can therefore disrupt their structure, causing the protein to denature. So if there's too many of these OHs at high pH, or too many of these positive H pluses at low pH, either one can denature an enzyme. Lastly, the effect of substrate concentration on enzyme activity. Again, let's use the analogy of enzymes are like construction workers. Well, they're building something. So what they do is they take substrate and they build a product. Let's consider that a construction worker, its substrate is nails. So as we are, say, down here, and the substrate or nails concentration is very low, basically the construction worker is just running out of nails all the time. The nails one nail into a board, and then is kind of having to look around for more nails, and so there's all this downtime for the construction worker. Same thing for for an enzyme, the yellow fellow, the enzyme here. The enzyme may bind to its substrate and catalyze a reaction, but then the concentration of substrate is so low that it takes a while for another substrate molecule to randomly diffuse into the active site and then have another reaction get catalyzed. So initially, as we increase the rate of substrate concentration, here we are, here's the description. Increasing the substrate concentration increases the rate of reaction. The construction workers are saying, finally, there's enough nails. I can just fill up my pockets and bang, 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 bang. And construction proceeds more and more rapidly as you add more nails, and the construction workers are not having to wait around for more nails. Same thing with the enzymes. As soon as one substrate is catalyzed and leaves the active site, another substrate molecule comes in right away because they're everywhere. There's a really high concentration. However, here, at the optimum concentration of substrate molecules, all the active sites are full and working at maximum efficiency. So any increase in concentration beyond the optimum will have no added effect, as there are no extra active sites to be used. Same thing with the construction worker analogy. If you're a construction worker right here, your pockets are full of nails. You've got extra pockets attached to a belt that's also full of nails. And then a dump truck comes along and dumps a big pile of nails in the yard. It's not going to make you work any faster. You have enough. So further increase in substrate concentration, no increase in rate of reaction. We've already reached the maximum rate. So essentially what's going on here is the enzymes are simply catalyzing reactions as rapidly as their structure makes them capable of doing. Here we have the effective enzyme concentration on the x-axis and again rate on the y. Think of enzymes as construction workers. Well, you add more construction workers, basically the building is just going to get built faster and faster and faster. And essentially on exams, on test books, this is how the graph looks, all right? Now, if you just took, I, for example, have, weirdly, a powdered enzyme, and if I just put all of the powdered enzyme into the water such that there was no water at all, I mean, it was just all enzymes, yeah, of course, there's eventually going to be a plateau here, but we, in naturally occurring biological conditions, this never occurs. There is never so many enzymes that it's going to be like all enzymes, no water, no substrate. Never happens. So basically, as the concentration of enzymes increases, the rate of reaction increases continuously. It's going to just keep going up and up and up and up and up. Enzymes can be inhibited by other molecules, and there are two types of enzyme inhibition. Inhibition can be competitive or non-competitive. Let's look at competitive inhibition first. So let's take a substrate. Ignore this right now, okay? We're going to ignore that. Take a substrate, and we're going to say that it binds to this enzyme because it fits the active site. The enzyme catalyzes the reaction. But no, there is this nasty inhibitor molecule here. And the inhibitor, if it is competitive inhibition, then we call it a competitive inhibitor, and it fits the active site and prevents the substrate from binding. So it is essentially competing to bind the active site. So therefore, the more competitive inhibitor molecules there are, the more often they will bind to the active site than the substrate, thus preventing the substrate from binding, reducing the rate of reaction. Okay, now let's come back and take a look at non-competitive inhibition. So now we're going to ignore all this stuff over there, and we're going to look at, again, the same kind of idea, same substrate even, same enzyme, but in this case we have an inhibitor molecule binding at a different site. These are also called allosteric inhibitors. Allo means other, and that's the root word. Steric means site. So another site. This inhibitor molecule will bind to this other site and causes a conformational shape change in the active site. So binding, let me erase that, there we go. Binding here changes the overall structure of the enzyme. So you can see that the active site itself, its structure has also changed, so the substrate cannot bind. So this really inhibits the rate of reaction. And the competitive inhibitor can also be taken off, either by other enzymes, or sometimes it is just not very stable and will spontaneously dissociate from the enzyme. That's true for competitive inhibition as well. Competitive inhibition revisited. We're now interested in looking at how different concentrations of inhibitor molecules affect enzyme activity, or I could say rate of reaction. So let's look at the first one. As substrate concentration increases, at zero, this first one, at 0% inhibitor, we get a normal curve. Here we go. Substrate concentration is increasing, rate is increasing, till we reach our maximum rate, at which point all active sites are continuously full, working at maximum capacity. No further addition of substrates has an effect. Okay. Now, if we then look at 25% inhibitor. So why did I choose 25? I just need more. So let's say 25 is right here, 25%. So 25%, you can notice at this concentration, when we have an inhibitor, the rate of reaction is lower than if we had no inhibitor. It's quite a lot lower in this case. But if we keep adding substrate concentration to this solution, or in a different situation, if we have more substrate available, basically what happens is eventually you have so much substrate there that the substrate is constantly outcompeting the inhibitor molecule for fitting into the active site. It would be kind of like somebody's giving away free tickets at a concert. There's one person with a red shirt there. There's one person with a blue shirt there. It's a 50-50 shot for who's going to get it, right? They're competing for it. But what if there's one person with a red shirt and 50,000 people with a blue shirt? Well, my bet is that the blue shirt, somebody in a blue shirt is going to get those free tickets, randomly simply because they'll randomly bump into the ticket giver or the active site, and they will occupy the active site. I hope that analogy makes sense. At 50% inhibitor, it is more inhibition. Here's 50% here, so you can see even more inhibition. And then as you add substrate concentration or increase substrate concentration, we can eventually reach the same max rate. Same max rate is attainable. It's still attainable because we can just add more substrate. This guy, we can just add more substrate to the point where there's just randomly, it's going to be much more likely for the substrate to fit into the active site. So a few more notes that are written on this slide. The higher the concentration of the inhibitor, slower the rate of the reaction. And even with competitive inhibition, the same max rate, as I just said this, will be achieved if more substrate is added. Because we have not changed the number of enzymes available, they're still going to be working, and their active sites will be occupied by substrate. Non-competitive inhibition is a different story because so again, here's 0% inhibitor, and there's our curve. So no inhibitor present, that's the curve. And as soon as we add some of these non-competitive inhibitors, they bind, as I said, to an allosteric site, a different site. So once the inhibitor molecule has bound, it is impossible for the substrate to bind, no matter how much of it there is. So you could have one substrate molecule, or you could have 10,000 substrate molecules. None of them can bind. And then if you increase the concentration even more, then we further reduce the maximum rate. So the reduction in max rate of reaction for non-competitive inhibitors is absolute. You cannot increase you cannot attain the same rate of maximum rate of reaction with no inhibitor if there is an inhibitor present, no matter how much substrate you add. Last thing here, and I'm going to let you guys take a look at this. I'll upload the PPT onto Blackboard, and essentially what happens here, this is a biochemical pathway. I might add a few more links actually, just to show you how all this works, but basically you have one substrate 1, that fits into this site, and it is going to make a product. We'll call that product 1. Product 1 binds to this active site, so we could almost call this like substrate 2 even, substrate 2. And then this enzyme produces another product. It's kind of like an assembly line in a factory, and this product fits into this active site, and then finally and we get this final product. We get a final product. And this final product is a molecule. We'll call it a molecule that is shaped like this. And this molecule will bind to an allosteric site on the enzyme that is the first one, so right here. And it will shut down this enzyme. It'll stop it from working, and that saves the cell from wasting energy, i.e., when enough of this final product here has been produced, it inhibits itself. It inhibits itself. And this is feedback inhibition, sometimes also called end product inhibition. And we can also, so many different terms, this is also an example of negative feedback control, meaning negative feedback. The feedback is this. Once we have enough of this product, the feedback is negative. It says no more. It shuts everything down. All right, that is all for this lecture. It's a long one. And remember, each week, if you were actually in class, you'd be attending 2 80-minute lectures. So it's really kind of about right on target as well. Just kind of depends on how you view it all. Hope it all made sense to you, and I'll see you on the discussion board.